The way I like to think about it in AML is right now we have really 5 main pillars that we're focusing on at MD Anderson. I think everybody else is focusing on in other major institutions. So the one I think is the FLT3 inhibitors. I think these are already here and they're here to stay. We already have midostaurin approved in the frontline setting. We're eagerly waiting for the results of quizartinib and gilteritinib. Both phase 3s have been completed a few months ago. Based on our experience in phase 2, we enrolled a lot of patients at MD Anderson. We really think both of these are going to be approved. We hope they are, and this will give us many choices, just like in CML, where we have imatinib, dasatinib, nilotinib. We would love to have 3 or 4 FLT3 inhibitors. If one fails, use the other, and so maybe you can increase survival 2 to 3 years. The second one, I think, are the IDH inhibitors. We've got the IDH2 inhibitor as single agent. I think as a single agent, they have activity. Their CR rate is 20%, overall response rate is 35%. It's not a huge blockbuster change, but again, I don't think they're going to be used as single agents 3 to 4 years from now. I think what we're really looking for is their data in the combinations, where there's early signals that were presented at ASH showing that both with azacitidine and 3+7, they may enhance response rates. And now we're actually starting trials combining them with targeted therapies. We have an IDH with venetoclax. We have an IDH with FLT3 that I'm going to be starting with the quizartinib. So we're looking at these, and these are quite exciting approaches, because now we're moving from the single molecular to combining molecular, maybe then adding AZA, and then getting better response. The third one, I think, that's really very important right now are the monoclonal antibodies. And it's very good that gemtuzumab, bosogomycin, the conjugated CD33, has come back to the US market. And then there are many others. There is a CD33 and a CD123 from Immunogen. There was some early data with the CD33 showing very good activity and potentially more safe than Mylotarg because of the concerns of VOD, but it remains to be seen. It's a phase 1. And then there are the bispecific antibodies, which I think for the first time at ASH, we saw some positive data with the macrogenics bispecific showing 5 out of 12 responses in relapsed, heavily treated patients as a single agent. Now, again, like I said with immune checkpoints, there are toxicities. This is not something you can do in a small community setting. You cannot do this outpatient. You need to monitor for cytokine release. But in the end, if you have a 3rd or 4th salvage young AML patient who has no options and you have a chance of getting a 35-40% response, I think that's where the bispecifics will come in. So after the antibodies, I think the 4th one is venetoclax, which is, of course, one of the major players. We think is going to be added to multiple regimens. It's already doing that. The frontline data, AZA venetoclax, we're one of the lead sites for that, is looking very good, as well as the low-dose Ara-C venetoclax, although the AZA venetoclax seems to be a little bit more potent right now with the long-term survival. And then I also presented some data at ASH and combinations of novel agents with venetoclax. So we had an MDM2 inhibitor, idasanutlin, with venetoclax, which in relapsed elderly AML population, where this is the worst of the worst people, you expect response rates of 10% or less, we were getting almost 50% CR/CRi at the expected dose. It's a phase 1, so it's early, but that study is being expanded. And if that holds true, then of course, can we move it to frontline using venetoclax MDM2 with low-dose AZA? So I think venetoclax is going to be used also with the immune checkpoints, with the antibodies. Pretty much it may become a way to use non-chemotherapies in the younger patient population with AZA, venetoclax, and monoclonals or immune therapies, giving potentially equivalent response rates. 75% is what you see with high-dose 3+7, FLAG-IDA. So that's really what we're hoping to achieve with that. And then the 5th one is the immune checkpoints. We think, again, these are going to be on their own as single agents, have very limited no activity, but are very good potentiators to improve durability of responses. So they can be added either in patients who are in remission or upfront with HMA plus venetoclax or HMA plus antibody. So I think we're really in this arena, just like multiple myeloma is the best example, where there's so many combinations, and now we really need to design smart trials. And that's really one of our efforts. We just launched a trial last month where I'm the PI. This is in collaboration with Pfizer, with 6 different arms, actually 7 different arms, where we're using each of their drugs, but in small cohorts of 15 patients each, with safety and efficacy endpoints that will encourage us or discourage us to move forward. So we're going to enroll, these include monoclonal antibodies, immune checkpoints, hypomethylating agents, targeted therapies, all in different combinations. And then once we get to 15 patients in each, we'll say 1 or 2 arms look really good, let's expand them to 40, and then if it looks good, Pfizer takes it and builds it. And I think we're going to have to move into this, because otherwise realize there could be 100 plus combinations, and it's not going to be possible to do an IST with 60 patients in each. And that's what lymphoma and solid tumor are doing, and that's kind of where we're heading.